Hello students, a very good afternoon. So before we start today's session, so I'm going to launch some technical polls. Okay. I'm sorry, we don't have technical polls today. So can you just tell me if my voice and my audio is clear for you? Okay. If my screen and if my audio and video is clear, please let me know. And if there is any problem with any of this, you can just put it in the chat box. Okay. So that would be really helpful. Yeah. So we have a minute. Please let me know if my audio is clear as well as my video and the screen is visible. Please let me know in the chat box. You just have to write yes or no. Okay. So that we can start our session today. Okay. So before we start photosynthesis, in the last session, we were left out with some of the important topics from biomolecules. So it's around 3 to 4 slides. We will cover up that small topics that are left out. And then we will start an introduction with photosynthesis, and we'll see what are the exciting topics that we have for us today with respect to plant physiology. So when it comes to some of the important aspects that we are talking about, enzyme substrate reactions, right? So we were talking about enzymatic reactions in biomolecules. So we were talking about how substrate concentration is very important for the enzymatic reactions to take place, isn't it? So one of those equations or one of those constants that will determine the enzymatic reactions for us is something called as Michaelis constant, or it is also called as Michaelis-Menten constant. So what does this Michaelis-Menten constant indicate? So it talks about our enzymatic velocity of our enzymatic reaction based on our substrate concentration. Okay, so we can just simplify this entire equation in two different terminologies. If our Km constant is high, if our Km constant is high, okay, then what is happening is our enzymes have low affinity. Whenever our enzymes has low affinity, now what will happen? Our enzymatic reaction, that is our velocity of the reaction, is reduced, isn't it? Similarly, whenever we have low Km value, okay, it shows stronger affinity towards our substrate. So whenever our enzymes have stronger affinity towards our substrate, then the product will be produced higher, right? So basically, what does this equation mostly talk about? It talks about the velocity of our biochemical reaction based on our substrate concentration. So this constant actually takes several factors into consideration, and then it finally, by the end of our chemical reaction or a biochemical reaction, we will understand how much was the affinity between your substrate and your product. Okay, so these two things you have to remember: what is high Km value and what is low Km value. Okay, so the next topic we had to talk about is metabolism. So metabolism is a wonderful machinery that happens in our body. So in simple terms, what is metabolism? As I said in the beginning of biomolecules, that our living system has this exceptional capabilities to consume any, for example, we consume different types of foods which are highly complex in nature, correct? No? So when they are highly complex, now what do? We break them down into simpler products, and through simpler products we will again reconstruct them back into complex products for our growth, development, repair, okay, etc. So this entire biochemical process of construction and destruction together, okay, for our all biological functions, this is called as metabolism. So in metabolism, what happens? Say for example, if you talk about your glucose, right? So glucose is broken down into pyruvate. Okay, during that process you produce ATP, and then you also produce carbon dioxide. So in this case, you have used oxygen to break down glucose into pyruvate and gain energy. So this is called as your catabolic reactions. Correct? No? When you talk about anabolic reactions, we talked about structure of proteins, right? And in humans, we have very specific structure. We have different proteins. For every proteins, they require specific sequence of amino acids. Now, what does our body do? From whatever the food we have eaten, it will break down the proteins that are in the food into amino acids and further use the same amino acids for the construction of the proteins that we require. So this is the whole cycle of your metabolism. So during this metabolism, what happens is whenever you're breaking down complex into simpler forms, so you release a lot of energy. Whenever you have to construct something, you spend a lot of energy. So this is construction and destructive process, which involves liberation of energy and consumption of energy. So based on this property, you can just put the entire metabolism into one summary, right? So metabolism can be of two important steps. One is catabolism, other one is anabolism. What are we doing in catabolism? We have larger molecules, fibers, okay, white proteins, and you have lipids, okay, and you have sugars and carbohydrates. You break them down into smaller molecules, okay. Through this process, what do you gain? You gain energy. Similarly, you spend energy on small molecules to construct bigger molecules or larger molecules through a process of anabolism. So this catabolism plus anabolism will give rise to metabolism, right? So our body has this capacity to take smaller things and construct big, and take bigger things and break them down into smaller smaller subunits. Okay, so when we talk about examples of anabolic pathway, okay, constructive pathway, where does anabolism happens? Simple to complex, no? So where does this happen? For example, when we convert acetic acid to cholesterol, okay, cholesterol is a very large molecule. Acetic acid is a very small molecule. Similarly, amino acid, which are the monomers of proteins, right? We use amino acid for the construction of proteins. So these are the examples. And photosynthesis is brilliant example for anabolism, because you have to remember, we are going to study photosynthesis today. Next one, when it comes to catabolic reactions. So catabolic reactions, what happens? Complex are broken down to simple. For example, glucose is broken down to pyruvate or lactic acid through glycolysis, or respiration process where glucose, yeah, same thing, right? So this is about different types of two important criteria of metabolism. Okay, so whenever we have a metabolic reactions, so we get an intermediate products, right? By the end of the chemical reactions or during the chemical reactions, we tend to get some products, and these products that are the resultant of your metabolism, that we call it as metabolites. Okay, so what happens for any substance or any compound to be formed? For example, if your body wants to, okay, use the glucose, okay, to produce energy, or if your body wants to construct proteins, okay, for some complex functions. So for any type of reactions that you consider in your body, it is not one step process. It is a very elaborate biochemicals, it is a step by step cyclic process. Okay, so our body is in a constant state of traffic. We can call it as a complete metabolic traffic. I can tell you, there are like hundreds and thousands of these metabolic byproducts that are just floating in your body without causing you any of this, okay, harm to your body. Imagine mixing so many chemicals into one system. How would a system react? But your body remains very stable. So our body is a constant, okay, our body, what it does is, it is between a constant fight of getting things into balance, or attaining equilibrium and going away from equilibrium. So what is the meaning of it? So if I have to create a system, for example, where one molecule has to be exchanged between one cell to another cell. So if there is equilibrium between two cells, do you think the movement of the substances will happen? Definitely no, right? So our body do not require equilibrium, and our body do not want to reach extreme states like extreme pH, extreme temperature, or extreme acidity. It does not allow that. So what our body does is, it will balance between both balance and imbalancing things, right? So it is trying to handle both of the things simultaneously. That is why we call our body as dynamic state of body. Okay, this condition is called as dynamic state of body, where all this molecular traffic is very well balanced, right, without having a severe reaction between each other, or without leading to any catastrophes. So as I was talking about metabolites, which are the resultant of metabolism, first we have your primary metabolites. I think we have already discussed about primary metabolites, that is your proteins, lipids, polysaccharides, okay, etc. When it comes to secondary metabolites are, some of the what do you call, resultant of these metabolic reactions. For example, it might be pigments, alkaloids. Usually in plant it happens, terpenoids, essential oils, toxins, okay, lectins, drugs, and then polymeric substances. So these are called as secondary metabolites. And why do we call them as secondary metabolites? Is because they are not strictly essential for the survival of the organism. When it comes to primary metabolites, it is 100% must. Okay, it has to be there in a living system. Only then a living organism can function. Accessory productions, whatever the cell produces, especially in plants, we can see all of this. Some of them are very economically important. For example, rubber. Okay, we have rubber, right? And for us, or we can talk about some morphine, which has medical properties. Okay, or lemon, which is used as scent. Okay, all of these are produced by plants, which are secondary metabolites. Even if they are not there, the plant can have a happy life without any worry. So now we are stepping into another chapter where we will discuss about photosynthesis, right? So we have a question before we talk about photosynthesis. So we don't have MCQs, that is, we don't have polls. So you just give the option, okay? Oxygen is not produced during photosynthesis by which of the following organisms? Okay, green sulfur bacteria, nostoc, cycas, and cara. So these are your options. So take a minute and try to answer in your chat box. Okay, just put a option in your chat box, so I'll follow. Read the question carefully. Photosynthesis is not produced. Okay, we have an answer. A, green sulfur bacteria. Okay, JNV Pune students have answered it as A. So I will give 10 more seconds for other students. So try to answer this. Okay, others. Okay, do we have other answers? No, it's fine, no problem. Okay, but try to participate, students. Okay, it will be very helpful for you. And or if you have any problems, if I am not aware, it's fine. Okay, so our question is, oxygen is not produced during the photosynthesis by which of the following organisms? So we have nostoc, cycas, cara, and then we have green sulfur bacteria. So our Pune students have answered green sulfur bacteria as the option, right? Correct option. So they are absolutely right. So what happens is, a photosynthesis can be of two types, right? So photosynthesis happens by reduction of for example, if you take water or if you take H2S, okay? So you need a proton in photosynthesis. So basically energy is used to split the bond between these two atoms, I can tell you, okay? So in nostoc, cycas, and cara, so they use water. Okay, they use water for the process of photosynthesis. They reduce water for the process of photosynthesis. Whereas when it goes to green sulfur bacteria, so the green sulfur bacteria, they live in acidic environments where they try to reduce something called as H2S or sulfur dioxide. Okay, that is why they do not release basically oxygen. They release like H2S they use, and then they release SO2 as the byproduct, not O2. Okay, so our option is A. So next one, you have heard about chlorophyll. So here is one question with respect to chlorophyll. The core metal of chlorophyll is, you have four options: nickel, copper, iron, and magnesium. So you have 1 minute to solve this question. So please try to answer in your chat box. So already we have an answer. JNV Pune. So they have answered magnesium. Okay, what about others? All right, students, we'll try to push forward. Okay, so when you look at the structure of chlorophyll, okay, magnesium becomes one of the important, right, important metals or ions to be present in your chlorophyll. So it has a significant role to play also. Okay, we'll understand by it. So for now we have an understanding, right? So it's not any other metals. So it is magnesium 2 plus or Mg2 plus is present in chlorophyll. All right. So let's go with respect to the outline of this chapter. By the end of this session, basically we'll try to finish how much ever topics as possible. So we aim to finish basically introduction to photosynthesis and how photosynthesis was proved through early experiments and how it gave our way of understanding photosynthesis. Next one is where the photosynthesis happens and pigments responsible for the photosynthesis to take place. And then we go and understand what is light reaction. And then in light reactions, we have two different types. One is cyclic and non-cyclic, okay, photophosphorylation reactions. And then we go for chemo-osmotic hypothesis. And then finally end with Kelvin cycle, okay, C4 and CAM pathways and photorespiration. So basically we will end our session with cyclic and non-cyclic photophosphorylation. But the entire chapter includes all these aspects like till the factors affecting the photosynthesis. Okay, so let us go for understanding what is photosynthesis. So since our younger classes, we've been listening to this that plants are autotrophs, so they synthesize their own food, okay, by using sunlight. So now also we have a little bit of idea about photosynthesis, right? So when we talk about photosynthesis, we talk about carbon dioxide and water being used for the production of glucose, okay, and oxygen as the byproducts, right? So by the end of the photosynthesis. Now, we are absolutely right in understanding this. So when we talk about photosynthesis, where do photosynthesis take place literally? So photosynthesis is strictly a light reactions, okay? So it is dependent upon light, okay? Part of a photosynthesis is strictly depending upon light. So it has to happen in the presence of light. So that is one of the criteria of photosynthesis. Second one, it happens in all of the greeny parts of your plants. For example, young stems, okay, apart from leaves, we are talking about young stems, okay, where it is still fleshy and there is a lot of chlorophyll, the photosynthesis can be seen there. So when we talk about photosynthesis, why do we even have to learn about photosynthesis? It is because of two reasons. So if you look at our biosphere, right, so we cannot survive without an energy cycle. So some or the other way, every single organism is dependent upon food on each other, right? Either through heterotrophic or autotrophic or saprophytic. You have different modes of nutrition. But when it comes to plants, plants are, okay, I will try to change the ink. Hold on. Okay, 1 second, people. Yeah, I've chosen black. If it is not visible, you just let me know. Okay, the first reason why we have to study photosynthesis is because, see, if we look at plants, they are producers, right? So producers are sorry, so they're 1 second, people. So they are producers, right? So when I tell about what is happening, excuse me, students. Yeah, it's gone. Okay, when I talk about production, so what is the role of it? That means there is no energy, right? There is no energy without plants, correct? No. So plants use very strict abiotic resources like sunlight, water, and the minerals that are present in the environment, and then they construct a very primary source of energy to our entire biosphere, I can tell. So since plants have this capacity to use the sunlight as an energy source to synthesize something called something in the form of carbohydrates or glucose, okay, that has given the way for so much of life to evolve, right? So without this energy cycle, there would be no heterotrophs. Without autotrophs, there are no heterotrophs. So without heterotrophs, there would be no such elaborate ecosystems that we have today. Okay, it would be only plants. All right. So life has evolved very much, and for the life to be responsible, for the life to be happening on this planet, that energy that is required for the life to be happening was given by our plants. So that is why we credit photosynthesis very highly. And second reason why we have to study photosynthesis is, so if you look at, apart from the plants, all living organisms which are aerobic, we utilize oxygen for our respiration, and it is must for our survival. So plants produce all the oxygen that we breathe, right? So for our respiration process. So these are the two important reasons why we start studying photosynthesis. So we have too much knowledge about photosynthesis because we have conducted a lot of experiments in our younger classes. So if you remember our younger classes experiments, so those are not like simple experiments that we did for exhibition. These were some of the game-changing experiments in our past. For example, let us look at the historical perspective of how people viewed photosynthesis. So if we take Joseph, if we take Joseph Pursley, he's a brilliant scientist, okay, or a thinker, you can tell. So Joseph Pursley designed this experiment where he would prove that plants utilize carbon dioxide and release oxygen, and he did it by using something called as bell jars. So imagine a jar that is of bell shape, okay, something like this. So he uses this jar, and then in this jar he'll light a candle, okay. In another jar he'll put a mouse, okay. He'll put a mouse. So both of them, like the candle will not burn for a longer time, and the mouse also will suffocate faster and it will die sooner. So what he does is he will try to put a mint plant, right? He'll try to put a mint plant in these jars. When he puts a mint plant in this jar, so now what happens? The mint uses this, carbon dioxide and releases this oxygen. So it will, elongate or prolong the life of this burning candle as well as the mice that is surviving in the jar. So that is how we'll be able to prove that, okay, the plants really use carbon dioxide and release oxygen, okay. And then using the same experiment, okay, we have Eganhouse, okay, where he talks about release of oxygen. So he says that only in the presence of sunlight, okay, only in the presence of sunlight you can expect the production of oxygen, he says, okay. So how did he prove that? So he uses the same experiment, but he uses aquatic plants. So in the jar, when he placed an aquatic plant, okay, so when the light was present, he could see the air bubbles around the leaves of these aquatic plants. So he understood that these bubbles that are produced by these leaves are nothing but oxygen molecules, okay. And further, what happens is, this is one of the revolutionary discoveries, I can tell, okay. Theodore, he gave an essential experiment where he proved that water is a very important ingredient for the photosynthesis to even take place, right? And then comes, okay, Julius Van Schaas. So here he talks about how plants produce energy and what type of energy do they produce, if they produce, right? So he says that plants produce glucose, and he doesn't stop there. He says that plants also have a mechanism like us where they store this glucose in the form of starch, okay. Then comes another important scientist, okay, there where we have Engelmann, okay. So here he tries to take something, prism, right? So what happens when we pass a light through prism? It splits into web jar or bands of light, right? So here what he says is, he wanted to know if all of the light is participating in photosynthesis or a specific part of a light is participating in photosynthesis. So when he split this spectrum, sorry, yeah, when he split this spectrum and he subjected the plants, so basically what happened is the plants started producing more oxygen in the presence of blue or red light. So that proved that a specific wavelength of light is really essential for photosynthesis, and not all the light is used for photosynthesis, okay. So, and finally, when it comes to Engelmann performs his experiments in a very keen sense. Like, he uses two organisms. For example, he uses a green algae, okay. He uses green algae, and then he puts them in suspension of aerobic, okay. So he puts them in suspension of aerobic bacteria. So he uses bacteria to analyze, okay, the utility of oxygen, and then he uses green algae to analyze the liberation of oxygen in this experimentation, okay. So, and then when it comes to Neil, okay, Neil actually performs an experiment where he proves that, okay, that photosynthesis is a light dependent reaction, okay. It is a light dependent reaction. So we require light for the process of photosynthesis to take place. And why do we require light here for the photosynthesis to take place? He also says that we require light to have hydrogen, or a proton, okay, from an oxidizable compound, such as you have water or H2S. These are called as oxidizable compounds. So we derive this hydrogen out of this oxidizable compounds like water or H2S, okay, and then we use this proton, okay, that we extract because of sunlight to reduce this carbon dioxide to form sugars, okay. So as he explains this, he also derives an empirical formula to explain photosynthesis. So today we know that we have 2H2O plus CO2 in the presence of sunlight, right? Sunlight. So we get C6H12O6 plus 2 molecules of oxygen plus water. Okay, if the balancing, I don't know about it. Okay, so for now you understood, like your glucose, oxygen and water will be the byproducts. Okay, so this is how the photosynthesis take place. Okay, so water is negligible here, right? So we know the reaction now. So but what he gave an empirical formula is for any type of photosynthetic reaction, like generally we don't take water. In some cases without water also photosynthesis take place. In that case we can use this empirical formula where we take is a H2S, okay, where H2S is an oxidizable compound, and in the presence of carbon dioxide we use sunlight to extract these protons in the form of H2 and reduce this glucose into some of the sugars like reduce the carbon dioxide into some of the sugars like glucose. So finally what happens is, after the empirical derivation of photosynthesis, Hill comes and proves that how the evolution, I mean like how the oxygen evolves during the light reaction. Like they talk about biochemical aspects of photosynthesis, all right. And finally Kevin comes and talks about the importance of carbon and how carbon is utilized during the photosynthetic reaction. So we have a famous name for it called as Kevin cycle. Okay, so this is about the historical perspectives on how people started understanding more and more. Like first they started talking about the release of oxygen and then importance of water, specificity of certain wavelengths, right? So empirical formula of photosynthesis, storage of glucose, in the form of sucrose. So all of the minor aspects that has compiled into greater knowledge and understanding how really plants manufacture food for the entire planet, okay, and for themselves also. So what is photosynthesis basically? So if you talk photosynthesis in a very plain or in a definitive sense, so photosynthesis can be just put as it is an physiochemical reaction, right? So it involves physical process also, it involves chemical reactions also. So if you combine both of them, it results in photosynthesis. So what happens in photosynthesis? We are using very small, I mean like the plants are using very small molecules like carbon dioxide and water. So by the end of the reaction, they are constructing something bigger. So we know that whenever we take smaller molecules and we construct bigger molecules, in metabolism, we call it as anabolic process. So our photosynthesis is an enzyme regulated anabolic process, right? It's a constructive process where we use simpler compounds and build complex organic compounds within the plant structure called as chlorophyll, right? In the plant structures where they have chlorophyll. So this is about photosynthesis. So it is a physiochemical reaction, or it is also called as anabolic process of manufacturing of organic compounds within the chlorophyll containing cells. So here you have a generic formula of photosynthesis where you use 6 molecules of carbon dioxide. Okay, my balancing was severely wrong. Here we have a balanced equation. So 6 molecules of carbon dioxide in the presence of 12 molecules of water. So in the presence of sunlight, chlorophyll and enzymes are really essential for the accomplishment of photosynthesis. Okay, that results in what? Our glucose, that is a sugar, and 6 molecules of water and 6 molecules of oxygen is liberated by the end of this chemical reaction. Okay, so one second. Yeah. So let us go to the further aspects of photosynthesis. So when we ask a question, where do photosynthesis take place? So it is very simple. Leaves are green and green leaves participate in the process of photosynthesis, and we have been learning about it for a very long time. So when we talked about cell structure and cell, the unit of life, on cell structure and cell structure and functions, we come across plastids and we understood the structure of chlorophyll. So generally, when we talk about chlorophyll, we talk about double membrane organelle, where it has outer membrane and inner membrane, and between the it is a space called as periplastid space, and within the inner membrane you have your stroma, and within the stroma you have some of the pillar like arrangement, like this, right? And these are called as thylakoids, right? So, oh sorry, grana, and the grana are made up of like small coin like structure called as, or coin like structure called as thylakoid, and each grana are connected by stromal lamella, right? And then we talked about thylakoid, how it has a membrane, and within the membrane of the thylakoid it has quantasomes, right? A special component which is essential for harvesting light. So these quantasomes have pigments called as chlorophyll pigments that participates in trapping sun's energy, right? We talked about chlorophyll. So chlorophyll is a region. We have chlorophyll, and we can divide chlorophyll into several regions. What are those regions? One we can divide as grana regions, and second we can call it as stromal lamellar region, and then we have stroma region, right? So what is happening in different regions of your chloroplast? In one region, for example, if you take your thylakoids or if your grana region, this is the region where your chloroplast will trap the light, use the energy that is coming from the light to conduct some reaction, okay, biochemical reaction called as light reactions. So the reason why we call it as light reaction is because it happens only in the presence of light. Okay, so what is happening in this light reaction? During the process of this light reaction, which is happening in our grana, like the chloroplast grana, ATP, okay, which is an energy molecule, and NADPH are produced as a byproducts of light reaction. Okay, so that happens where it happens in your grana region. When it comes to stroma, the matrix of stroma is where all the enzymatic reactions happen without the dependency of light. There is no light requirement here, and it happens strictly because of the enzymatic, okay, the enzymes are highly involved in this process. So which in turn, by the end of this reaction, it results in the formation of starch or energy product or carbohydrates. Okay, so since it happens without the dependency of energy, so this we call it as dark reactions. So we understood what is light reaction and what is dark reaction. So both this light reaction and dark reactions are happening within the chloroplast. So in what regions of chloroplast? You have your grana, which contains thylakoids. Thylakoids have pigments. So pigments are meant to trap light. So when they trap light, they undergo certain process or reactions. So these reactions which happen in the presence of light are called as light reactions. And the resultants of these light reactions are ATP and NADPH, right? And now that is happening in your grana. Whereas when it comes to stroma, it have enzymatic reactions that results in the formation of starch without the dependency of light. So that we call it as dark reactions. Okay, so just because we call dark reactions, that doesn't mean that these dark reactions do not take place, okay, when the sunlight is present. It happens in the presence of sunlight, but it is not dependent on the light. Okay, that we have to remember. So for this entire process to begin, for the photosynthesis to even begin, initially we have our photosynthetic pigments. Okay, that is the foundation, isn't it? So we need light to harvest something called as protons. So we need energy to reduce our carbon dioxide into sugars. So to this process, what happens is we have very special pigments. Different types of pigments are there within our leaves. So if I take a look at our leaves, right, our leaves are of several colors. Sometimes they look yellow, orange, okay, greenish, and little more brownish color leaves are also visible. So why is it? Because we think that only green color is chloroplast. Actually, we are wrong. Okay, the very important type of chloroplast is bluish green. Okay, and the next important chloroplast is yellowish green pigment. So it's not essentially it has to be green pigment. Okay, so these pigments are of chlorophylls are of two types. One is chlorophyll A and chlorophyll B. And chlorophyll A is bluish green pigment, whereas chlorophyll B is yellowish green pigment. So now this chlorophyll A is very important pigment for our photosynthetic reactions, and we'll learn about it. So there are accessory pigments. So when it comes to chlorophyll B, also it is called as accessory pigment. Xanthophylls, okay, and keratin, and then we have phenophytin. So these are accessory pigments that are present in our plastids like chloroplast, which help in our harvesting extra amount of light. So the pigments are present in our chlorophyll for one purpose, that is to harvest light. So if you talk about chlorophyll A and chlorophyll B, so they are highly specialized in absorbing or trapping one specific wavelength of light, which usually appear in blue or blue and green, blue and red colors. So when you talk about carotenoid pigments absorb, okay, some of the, we can call it as lower wavelength light, I can tell you. So something like purple lights, okay, or the purple color. So why do all these pigments are necessary for harvesting? If we have only chlorophyll A, then our absorption of light becomes very less narrow. But if you have more pigments which are capable of absorbing a lot of different energies or different wavelengths, then the net absorption of the light is higher, isn't it? So this is about photosynthetic pigments. So what we do is we subject something called as chromatography, okay, where we dissolve all these pigments. We take paste and then we extract the filtrate out of this leaf paste, and then we put a solvent and put a chromatographic paper. So when you put a chromatography paper, now the migration happens based on the molecular density of the pigments. Okay, it divides in the form of colors, different colors. So by recognizing the type of color, we can mention how much chlorophyll A or what are the different pigments that are present in a specific plants. Okay, so you can see here very easily what are the different pigments and what is the role of chlorophyll. So we just talked about it. So this is about chromatography I just talked about in the left end of your image. So where the separation happens based on the color, and that is basically on the composition of the pigments. Okay, so next let us go for the next one. So do pigments absorb all the light? And we talked about it. So when it comes to photosensitive wavelengths or photoactive wavelengths, so there are specific wavelengths in which our pigments react very positively or very highly. So these pigments these lights are between blue and red color regions of our spectrum. So here we can see how chlorophyll A, which is one of the chief pigment, it will associate with photosynthesis when a particular wavelength of light is used. So you can see here. So if you see your chlorophyll A, right? Chlorophyll A is very much okay with the pigments such as 680 to 700 nanometers. So you can see how it peaks, right? Its absorption or the light harvesting capacity becomes very high between 600 to 700. We need higher wavelength, right, with respect to chlorophyll A. But if you look at the accessory pigments, they can absorb lower frequency of light, like 400 nanometer. You can see some of the pigments can go up to between 400 to 500 nanometer. So if you take about chlorophyll B, it comes between 400 to 500 nanometer. So they require lower wavelength. They can absorb lower wavelength of light, right? So another graph, it shows about rate of photosynthesis with respect to, again, wavelengths. So when we have specific wavelengths, the resultant of photosynthesis is very high. So for example, when you are it is very cloudy or very foggy, okay, we cannot expect the same photosynthetic by a product like in a very normal bright day. Or if it is extremely hot, okay, or the temperature is very high and the light is too bright, then also the photosynthesis will not be at the peak levels, right? So this is about the specific wavelengths our photosynthetic pigments use, okay, for the purpose of photosynthesis. Fine. So on the next thing we have to study about is photosystems based on the arrangement of your pigments, right? We talked about chlorophyll A, chlorophyll B, and other accessory pigments, right? So they organize themselves into two different photosystems based on their capacity, on observing a specific wavelength of light. So that we can determine as photosystem 1 and photosystem 2. So basically we call this system or the organization of these pigments as photosystem 1 and photosystem 2, not because they are high priority based on their function. So it is based on the discovery. First the photosystem 1 was discovered, later photosystem 2 was discovered. Okay, it is not in the on how they react also. Okay, it is just this was discovered first and the next it was discovered, so they just named it. But when it comes to reaction, it is quite different. Okay, so we should not correlate these two things. So when we talk about photosystem 1 and photosystem 2, give me a second, people. Okay, so we will talk about photosystems in the next class. Okay, so if you had any problems with this session where you didn't understand the concept, or if you have any doubts, or like NPS, right, like doubt solving or delivery or content problem or anything is there with respect to the session, you can just put a message. Okay, we have 1 minute to end the session, so you can do it quickly, people. Okay, that would be very helpful for me. And students, I'll clarify, we'll continue this in the next class so that we will have proper understanding in learning about more reactions. We can't just put it half and half. All right, we'll end the class here, and please give me a review about if you had any problems with respect to the session. Okay. Students, could you make it fast? Probably you will be late for the other classes too. Okay. By the time you do this, I will check out our YouTube friends. Okay. So let's see how it's going here. Okay. All right. So there are no chat or any doubts here in YouTube also. So fine, students. Okay. We are out of time. We can talk more about photosystems in the next class. Okay. Thank you very much. See you. See you in the next class tomorrow.